Hi, we are team number 5 and we will present the topic of translation. My teammates are Mariana, Dalia, Paloma,
Daniel.
Gonzalo.
This is the ribosome. Then we have the anticodon. Then we have the codon.
This is the tRNA, and this is the amino acid.
And finally, this is the protein chain. Translation is a process where it is necessary to transfer the message from a language that is in terms of nucleotides to another in terms of amino acids. In other words, it consists of the decoding of an mRNA sequence into a protein. The key to understand this process lies in codons, where each sequence of 3 consecutive nucleotides goes for a specific amino acid.
The genetic code is the set of rules used by living cells to translate information encoded within DNA or mRNA sequences into proteins. It consists of 3-letter combinations of nucleotides known as codons, which map specifically to one of 20 amino acids or a stop signal during protein synthesis. tRNAs present 3 exposed bases called anticodons, which are complementary to the bases of a codon on messenger RNA. When the mRNA appears, both subunits join and hold it between them.
Ribosomes are the organelles where translation occurs. They are composed of 2 subunits, one large and one small. Each is formed by proteins and ribosomal RNA. The large subunit has 2 binding sites for transfer RNA and one catalytic site, where the peptide bonds forms between the amino acid carried by the tRNA molecules.
The first step of this process is initiation. This begins when the mature mRNA leaves the nucleus and reaches the cytoplasm. Here, the mRNA joins a small subunit of the ribosome, then a specific tRNA arrives carrying the amino acid methionine, and this is paired with the start codon of the mRNA. Finally, the large subunit joins the small one and closes around the mRNA.
Elongation. A second tRNA carrying another amino acid arrives and its anticodon pairs with the corresponding codon of the mRNA. The first amino acid, methylamine, detaches from its tRNA and forms a peptide bond with the new amino acid. The tRNA that has already released its amino acid exits the ribosome, allowing new tRNAs to arrive. Their amino acid bond through peptide links into correct sequence, forming a protein. Termination. It occurs when the ribosome reaches a stop codon on the mRNA. This codon pairs with a release factor that facilitates an enzymatic reaction, which in turn releases the mRNA and the ribosome protein.
Making proteins through a process called translation is one of the most important jobs in the human body. Through this process, our cells read the genetic instructions to make the proteins that keep us alive. As Lourdes et al. Noted, the translation of the genetic information into proteins is an essential process for all living organisms. Without this step, life would not exist at all. Translation is also vital in medicine, but if in translation occurs an error, diseases may occur. However, translation provides us tools like mRNA vaccines that help us to save lives. So overall, translation is a key process for all living organisms to keep us alive.